we can adapt accordingly. We've got some time and flexibility here. But I would like to welcome Leila Wehbe from Carnegie Mellon University, who is giving a guest talk today. Leila did her bachelor's at the American University of Beirut in electrical and computer engineering, and then came over our way and did her PhD in machine learning with Tom Mitchell, and focused a lot on machine learning approaches to decoding analysis on neuroimaging data. And then Leila went on to do a postdoc with Jack Gallant at UC Berkeley. So she's been at both CMU and UC Berkeley, which are two places very near and dear to my heart as well. And kept working on detailing complex decoding analyses on neuroimaging data, doing a lot of really phenomenal work in that space. And in 2018, came back to Pittsburgh and started as faculty in the School of Computer Science and Department of Machine Learning, really kind of bringing back all that expertise that she got over at Berkeley and expanding the kind of focus on what I call computational neuroimaging. And Leila's been doing incredible work in this space over the last few years as well. So please welcome Leila, and the floor is yours.
Yeah, thank you so much, Tim, for the kind introduction. And hi, everybody. I'm very excited to be here. I was thinking, just considering the format, it would be a bit more interesting to have a discussion. So I'll try to just stop and pause and ask for your comments throughout the talk. And that would probably mean, or especially if you have any questions, please do interrupt. And maybe that would mean that we will not go through everything in the talk, but maybe that's actually more useful because we would just discuss the more interesting or relevant things for you. So a lot of my work is about, or the main tool that I use in my work is building encoding models to study how or where and how different types of information are processed in the brain. And lately it's been quite exciting because of the kind of the rapid progress in deep learning. It's been quite exciting because now we have neural networks that encode a lot of the information that we're interested to study in the brain. And so, especially, for example, in the domain of language, which is the domain I do most of my work in, it's these models, because they are very powerful and they're able to express kind of the meaning, not just, or to come up with a representation, not just of the meaning of the individual words, but also of sequences of words. They've allowed us to really ask much more complex questions. So I'll just give you an example of the kind of questions I'm talking about, or the kind of things that we can study now with this interface between AI and neuroscience. And the interesting thing you will see is that these questions are not just neuroscientific or just AI questions, they're actually shared questions. And because basically both fields are studying the questions from a different angle, they can benefit a lot from interacting with each other. So one of them is, for instance, about whether or not, or how to build representations of information or of a stimulus that is task dependent or task invariant. For example, if you were to want to achieve something or to perform a task with a given stimulus, would you encode it differently based on the task you want to achieve, or would you still encode it in the same way and kind of do some higher level processing at some later point? Another question, which is also quite interesting and quite complex and kind of very core in this case to language and other fields as well, is a question of compositionality. So when we talk about compositionality in language, what we talk about, or what we think about, is how our words combine together in order to create meaning for sentences or for phrases. And the interesting thing here is that when you combine words together, you achieve a meaning that is actually superior to, or different or beyond the meaning of the individual words you're combining. So just a few examples. If you think of the sentence, Alice finished the apple, you understand that this means that Alice finished eating the apple, even though the word eating is not in any of those words, or the meaning of eating is not necessarily in there. You also understand sentences like Alice gives Bob an apple and Bob gives Alice an apple to mean different things, even though it's exactly the same words. Another example of composition is when you say green banana. And here, the word green, when you combine it with banana, you get the concept of unripeness, which is not really there if you say something like green book. Okay, so you can see how these questions are quite complex. And if you want to answer them in the brain, you might want to study, you would have to study so many things at so many levels, from kind of how is the brain perceiving individual words, how is it combining them to form sentences, and also how is it incorporating reasoning in order to understand these sentences. Like, for example, for a lot of language processing is not just language, it's not this isolated domain that doesn't interact with other things. You also use things like spatial reasoning, or use your memory, or you use any kind of inferences in order to understand the world around you. And the good thing is that we kind of moved away from kind of the 19th century understanding of language as being, just confined to these two areas in the left hemisphere, to a more complex understanding of language. But unfortunately, we do not have one single understanding. If you think of the different models of language processing, you find that there's actually a few of them. And even though those few of them kind of more or less target the same areas, which are the left, maybe temporal and inferior frontal cortex, they actually disagree a lot on what these different regions are doing, what they are processing. And I think that one of the reasons for these disagreements is that these models are built from individual experiments in which one of those things that underlies language is studied at a time. So you take this complex thing that is dynamic and has all these things that are moving together. For example, in language, you have syntax and semantics, and you have anticipation of the next word. You have all of these processes are going at the same time. And when you run a classical language experiment, what you have to do is you have to kind of fix all of those variations and just vary one at a time. And when you do that, it's not clear that you would necessarily get a good model of how all these things are varying together. And so the alternative approach to doing this is to run a naturalistic experiment. So this is an experiment of this is an example of an experiment I ran at CMU, where people sit in the scanner and read Harry Potter. So here, one word is presented at a time to the subject, and the fMRI machine is recording at a rate of 1 image every 2 seconds. And the idea here is that what we are going to do is we're going to try to answer any question. So you collect this experiment, but the nice thing about this experiment is that it allows you to ask any other question afterwards. Like, let's say you're interested in studying syntactic representation. You're going to try to build a model that goes from the words that the subject has read to the brain activity. So we can think of the brain activity at any given point in time as just a row of voxels. And another thing, which I'm not going to mention here, is that, of course, there's a delay between the words you read at a given time and the time that you the recording in the brain. And so here, in order to deal with this, there's multiple ways. One of the things we do is we kind of delay the features of the input stimulus in order to kind of capture the latency of the dynamic response, of the hemodynamic response. But we can talk about this separately. It's a detail at this point. So let's just assume for a second that the latency is taken care of. So now, again, I want to study syntactic information, like which region in the brain is representing syntactic information. And so one thing I can do is I can build a feature space. So this feature space will tell me at every point in time, such as this point, what syntactic information is present. So here, again, this is kind of a toy syntactic space that I just built for the sake of this example. And it has a few parts of speech. So it tells me like how many verbs, pronouns, articles, or nouns, etc., occurred at this particular point in time. And so now you can imagine labeling your entire experiment, every TR of your experiment, in terms of what has occurred at that time, and then trying to build a function that predicts the activity in each voxel independently when taken as input the feature space. So the idea is that the importance of learning a different function for every voxel is that this function is actually telling you what you, like it's actually estimating how these individual voxels are responding to the input features, which in this case are syntactic features. And so this is the broad kind of framework for this whole encoding approach, which is now becoming more or less established and common, where instead of going from the you replace your stimulus with a feature space, you build a predictive model. So here I just have the function f here, which is because it is agnostic. Like there's been a lot of work on trying to see whether this f function should be linear or not, whether it should be neural network or not, and we can talk about this too if that's a topic of interest. But I think really the most interesting part is this part, which is the feature space, because really like you can put any kind of feature representation of the stimulus here and test again another hypothesis and another hypothesis. So I told you how you train, how you would build this, but you also need to test it. So there are also multiple ways to test how good your predictive model is. The simplest way is to just take held out data, so data that was that the subject saw in the scanner, but that you did not use for training. You replace this text by its features. In this case, we have syntactic features. And then for each voxel, you predict the activity in that voxel from the features. And then you can compare the real data that was acquired at that voxel with the predicted data. And in some places in the brain, which don't care about syntax, you're not going to be able to predict well the brain activity. But maybe, hopefully, in the places that actually do care about syntax and specifically about parts of speech, you're going to, and if like everything went well, you have good features, you have a good model and like not too much noise, you hopefully will be able to predict well the real data. And one way you can compute this is by just computing correlation between the predicted and the real data. And of course, this is just an example. There's like 4 time points. In reality, the test data is much longer than that. And then you run the proper hypothesis test. So you need to determine that the correlation or any other prediction performance measure that you compute, you need to determine that it is actually significantly higher than chance. And of course, you need to correct for multiple comparisons because you have so many voxels, and maybe you're also trying multiple feature spaces. And then if at the end of all of this, you find regions that are that are significantly predicted by syntactic features, then this would suggest to you that this particular voxel in the brain is representing syntactic information. Now, of course, I mean, this is something that is plausible. However, if you've worked with any kind of natural stimulus that's not been controlled, you would have noticed that actually features of, like, for example, features of language are quite correlated with each other. So just because something is predicted by syntactic information, some area in the brain is predicted by syntactic information, it doesn't necessarily mean that it's representing syntax. It might represent something else which is correlated with syntactic information, like, for example, meaning, or word length, or frequency, something like that. But more or less, this is kind of the framework that we use as a tool in this work. And more recently, and you might have seen this already, but people have been using this to kind of compare representations in the brain and representations in neural network, just again using this fact that you can kind of substitute the feature space over here by any kind of representation. So because we have neural networks that are able to, for example, here convolutional neural networks over here are able to kind of take an image and kind of process it hierarchically, and actually these networks are actually inspired from findings in the visual system to start with. But because we have that, we can actually take the representations of these different layers and try to see which area of the brain do they predict. And we see a very nice alignment, or the field has seen a very nice alignment in vision between the different layers of a neural network and the different areas in the brain, the primate brain, I would say, both humans and monkeys. But you can also do the same thing with something like a language model. So here in language, we kind of more or less process input sequentially. So you need other types of models, for example, a recurrent neural network language model. And so this is a simple kind of such a model where basically at every point in time, the model integrates the current word with its representation of the previous context to generate the context for the next word, and from that predict what the next word might be. So we can just unroll this to see it more easily, and we can see that like at every point, at every word in the text that you have, or any text you're interested in, or any sentence or passage, at each step, this blue vector is representing basically everything that's been seen so far, and then the model sees this word, and once it knows what the current word is, it can see how well did it predict that word was going to appear at that point in time. But again, it can combine the previous context in blue with this magenta vector to predict the context at the next stage. And once it sees the word at the next stage, it can again verify its prediction, and we can also like kind of combine it again, et cetera, et cetera. So in some sense, this model is reading the text in the same way, more or less, that subjects that are sitting in the scanner are reading it. And so one interesting thing to do is to take basically these layers of the model and try to use them to predict brain activity. And this is something we've tried in MEG. And we've seen that both the context vector and the current word vector actually predict a large part of the MEG sensors and time points. And I'll say a bit more about that later. And others as well have tried to use other methods, like for example, EEG here and a recurrent neural network model for grammar to kind of recover basically these different ERPs that we know that are associated with grammatical processing and find this correspondence. And there's also this work by Shaley, which I think is on this call, in which also what was found is that if you use an LSTM model that is processing the language input in the same way that the subjects are, you can predict large parts of the cortex, including, again, this is bilateral, not just left hemisphere. And you can predict basically large parts of the temporal cortex and the frontal cortex, as well as additional association, additional parts of the associative cortex, or even other parts as well. And there's an interesting kind of differentiation between kind of regions that prefer short context or can be better predicted by short context that's incorporated in the model versus long context. And again, I just want to add one more point here, is that every time in my experience that I've seen people use naturalistic stimulus, we always see this kind of bilateral representation of language and not just left hemisphere representation. It's very symmetric. And that by itself is kind of a fascinating topic of study, like why do we have this very highly bilateral representation, whereas all the controlled experiments and also all the kind of lesion studies and all of the things that are not naturalistic experiments of language are very left lateralized. So, okay, so just as an interim stop here, I just told you basically how at a high level, how you would kind of do this kind of relationship or study or use neural networks to model brain activity. And again, as I said, this is becoming really a rapidly growing area. We have a conference now in like computational or cognitive computational neuroscience, in which a large part of the work is about this kind of relationship. We also mentioned in passing that actually there are problems which are, for example, that there's a lot of correlation between different features that you might want to estimate from the stimulus, which actually kind of obscure a little bit the conclusions that you might want to make. Another thing is that the neural networks themselves are actually hard to interpret. So just because you've kind of extracted a network or a layer from a network and try to predict the brain with it, and you find that it predicts well, that doesn't necessarily tell you a final answer of what's going on, because the original layer itself was hard to predict. And this is where it would be interesting to basically use certain approaches or certain types of controls. We're trying to call them now computational controls, in order to still be able to make scientific inferences using them. For example, in the work I mentioned before by Shaley, you can kind of see, you can manipulate the variance, or you can manipulate how many words you're inputting into the model to kind of study basically what is the optimal length of context for a specific area. And I think she has more recent work about that. And I'm also going to talk to you about kind of other ways we can do this. But maybe for now, I'd just like to stop and see if anyone has any questions.
I was going to ask, what's the primary objective in this? Is it to understand exactly how brain represents them, or which areas are firing up in what part of the language processing?
Yes. So yeah, for sure, one of the main goals is to kind of understand better how the brain is processing this kind of complex information that underlies these complex tasks. Of course, when you use something like fMRI, the questions that are easier to answer are the questions of where, like which areas are doing which or what part of the information. But again, because we're trying to do this modeling, we're not just answering where, but we're also answering in some sense what. So we are able to not just say these two areas are activated by the stimulus, we're able to say this area is predicted by these particular features of the stimulus, and this other area is predicted by this other feature of the stimulus. And once you have this kind of understanding, you might be able to maybe propose a better model of kind of how these areas are talking with each other. And another thing to also mention is that once you create, or once you use, for example, a neural network to study, or you relate a neural network with the brain activity, what you're saying is that actually this particular neural network is a model for the brain. It's kind of a proposed model for what the brain is doing. And of course, it's not going to be a perfect model. But one of the long-term goals is to eventually kind of just modify this model until it's kind of resembles the brain more and more. And one more goal, I would say, is also to kind of go the other way around. Actually, like this is over here. So the first one is kind of using AI methods in general to study the brain. And the second one, which the third one I just mentioned, is kind of like working together to like kind of make these AI models just better models of the brain. And the middle one is the one I just wanted to mention, which is not just kind of doing all of this to benefit our knowledge of the brain, but also maybe ask whether studying the brain in conjunction with these networks, or in conjunction more generally with AI, will help us improve AI, and in this case, natural language processing or computer vision.
Thanks. I have another comment if nobody else has any questions. Yeah, so when you mentioned, because you highlighted the importance of exactly what feature space you look at influencing the results and understanding, I was thinking, has there been any study where they actually use multiple feature spaces, for example, touching upon both the syntax, meaning, emotion, and other things that are involved in naturalistic stimuli and language processing in, for example, say a multiple kernel learning framework, and see which type of features are actually predicting a particular region. I mean, I actually don't want to use the word predict, but is linked to a particular region or aspect of the how brain is processing the language information. So I think that would yeah, like, has it been done, or is there some utility in doing it?
Yeah, so I think that's a very, very interesting question. So I think definitely that's kind of the larger goal, I would say, or one of the goals. So in one of our papers from 2014, with this Harry Potter stimulus, we did try to model basically all of the different things at the same time. So so the semantic features of individual words, the syntactic properties of the words, the kind of the emotions that the subjects were, sorry, not the subject, that the characters were feeling, the characters you were reading about were feeling, and things like kind of, I don't know, actions and and some other things like that, as well as kind of more low-level things like basically how many words are in the screen. And if you model all of these things, what we did was model them separately. You find actually, especially for the ones that are more high level, like the discourse level information, like the emotions of the characters or even the identity of the characters, you find that they actually predict different parts of the brain. And also interestingly, but again, this is kind of more like a kind of a hypothesis. There's definitely much more work to do there to verify this hypothesis. But for whenever you read about characters engaging in communication with each other, you do have kind of basically the same region that you use in everyday life to interact with other people, to interpret other people's feelings, or to do theory of mind in general. The same region is predicted or is well predicted by these kinds of features when you're reading about people. So, and also when you kind of think about motion or read about motion of characters, the same region that you use in everyday life to perceive motions of others visually, you also get that same region well predicted by the motion in the text. And so kind of one way, one nice framework, and this is kind of supported by some other theories in language, like not in neuroscience, is that kind of reading is a bit something like an internal simulation of the world. And so you just use basically the same regions you use to interact with the world. But also you mentioned kind of some learning, like multiple kernel learning or something to that respect. So of course, from a machine learning point of view, just training or statistical point of view, just training individually with these different feature spaces might not be optimal. Really, you want to kind of combine them all together and try to basically come up with a model that kind of partitions the variance of that is explained among these features in some way. And there's definitely been work on that, both from us, from our lab, but also from others working on that. Anyone else has a question or a comment or?
No, I'm excited about the kind of next part to jump into. So
okay, great.
I'll restrain my comments.
Okay, sorry. Okay, so I'm just gonna talk about basically some examples of what I just said is, which is, even though this approach is not perfect, even though there's kind of correlations between different feature spaces, and even though maybe deep networks are not interpretable, we can still use them by kind of creating these kind of computational controls. And I'll show you some examples of some things we have done, and please interrupt if you have any questions. So the first one is kind of recent work with my student Maria and with Tom Mitchell, in which we, again, we're trying to think of basically where is composed meaning, the composed meaning of sentences represented, and how is it represented in the brain, especially in relation to the meaning of the individual words when taken in isolation. And so to do that, you can take a language model. In this case, we used Elmo. So it's a bit more complicated because it's a kind of a bidirectional language model. But if I just take this particular part of the network, which is just kind of the forward part, it's a bit similar to what I discussed before about a context vector. And here I'm taking basically the context before seeing the current word, which in this case is sent, as well as this vector, which does not have any context in it. It's just basically the vector that's attributed to the current word, or that it's like the embedding of the current word. And so when I use basically these two vectors to predict brain activity, I get, and this is an example result for one subject, I basically get all of these regions that are well predicted by both. So anything that's in white is well predicted by both this context or sequence vector and by the single word vector. Regions in red are only predicted by the sequence vector, and regions in blue are only predicted by the word vector. And here my prediction performance is actually a classification accuracy. And this is very similar to the correlation, they kind of both go up together and go lower together. It's basically a classification task that's on top of the predictive model. And again, this is a detail I can discuss later. And so, so you can see here that actually there's many regions, especially in the left, basically temporal and frontal cortex, that are well predicted by both the sequence model and the word level model. And you might think that actually this is because these regions are doing both the current word and the previous context. But if you've thought about language models before, or these models, you would think that actually it makes a lot of sense. Well, it's not a surprising result, because actually the context vector that's immediately before a word and the embedding vector for that word are necessarily very correlated with each other, because this model is trained to predict the word at a specific position. And even if you take, basically you try to predict, even if you're trying to predict basically the embedding of the word at these different locations, you'll see that actually if you take the context vector, you're able to predict the word at this particular location very well. So this is the red line is the context vector evaluated at t-1, and it's showing you all the words that this context vector can actually predict well. So there's an inherent correlation between this context vector and the current word vector, which will complicate our interpretation. And so here, this is one of the kind of computational controls that we do, in which we learn a function, g, such that this function can predict the context vector from the words that were used to compute the context vector. Okay, so we take the network, we input, I don't know, 20 words to compute the context vector. Here we're gonna do something else. We take those 20 words and we learn a linear function that can predict the encoding, the context vector from the individual word vectors. And so very crucially, this function is linear. And so what we're saying is that we're trying to kind of predict everything that can be predicted from those individual words linearly. And then we're going to take the residuals. So these are the residuals of the context after taking out everything that's predicted linearly from the individual words. And so again, going back to this notion of kind of combining words together to get a new meaning that's not included in the meaning of the individual words. This is our estimate of that, which is something we call supra-word meaning. So the supra-word meaning is something that's added by combining the words together that was not there in the individual words. And now what we can do is we can just go back to our predictive model and like take those context residuals and predict the brain activity. And we can do the same thing too for the words. We can also take the word residuals. And so, so we're kind of comparing this method to disentangling. Basically, we're trying to kind of capture information that's only in the context vector or only in the individual word vectors. And once we do this, we find that again, some regions, like this supra-word meaning, does predict regions in various parts of the language system. When we look across subjects, we find that actually, specifically in the anterior temporal lobe and the posterior temporal, we find that actually the context vector, the supra-word meaning, predicts reliably across many, across our subjects. Now I'll show you something else which is quite interesting. So we repeat this manipulation in MEG. So we have the same experiment in which subjects are reading one word at a time, but with MEG recordings. And so every word in our case lasts for 500 milliseconds, and I can take that time when a subject is reading an individual word, and I can try to predict the activity during that time using these different vectors that I have. And so I see here, so the light bars are when using the original embedding for word T, and the dark bars are when using the residual embeddings. And I can see that both the residual and the full embeddings actually predict well the activity when this word is being read. And so basically what this is saying is that information that is specific to a recurrent word is predictive of the activity when the word is on the screen, which makes sense. But also we see that when the word is on the screen, there's also information from the previous word that's predictive of the activity. And this is also true when we kind of regress out the shared variance with the current word. So, so basically, you can see the dark yellow vectors over here. Those are still, or yellow bars, they're still kind of significantly higher than chance. And what this is saying is that there's still information that's unique to the word that just appeared, the word T-1, that's predictive of the brain activity at this time. Now, the surprising part is over here. So once we remove from the context vector the information that's specific to the current word and the previous word, specifically those two words are the most important ones. Once you remove them, you actually are no longer able to use the context vector or the context information to predict anything basically during the current word. And this is surprising because it's not like these supraword meaning factors are not predictive of activity at all. They are very very predictive of fMRI activity. So there's something in the brain that is being captured by these vectors, but it's not, that something is not visible in MEG. Okay, so what does this, or what do we think this means? So here, in the case of fMRI, we think it's interesting, specifically that we can predict activity in the posterior temporal lobe from supraword meaning, because in many theories of language processing, the posterior temporal lobe is associated with kind of lexical meaning, or the meaning of individual words. And here, one nice hypothesis that's coming out of this is that maybe the meaning that you get from combining individual words together, this combinatorial meaning, is also present, or is also processed by the same brain structure. So that would be something nice to study in future work. From the MEG result, we think this is interesting. It's interesting because maybe what it's implying is that this process of maintaining information, or kind of combining information over multiple words and maintaining it, because it's not visible in MEG, it might be due to the fact that the brain mechanisms that support this integration, or not the integration, but the maintenance of this information over many words, maybe these brain processes are just not the kind of processes that lead to high MEG activity. And maybe, so basically what we're trying to say is that it's not due to kind of aligning, it's not due to neurons firing in synchrony, for example. And again, people have used MEG extensively to study things like composition. So we're not saying that you cannot use MEG to study composition or do anything like that. It's actually a very good tool for that. But we think what is present in MEG is not necessarily, is not just maintaining the information. It's more like, how is this information change, or how are you integrating new words into this information, and not necessarily this kind of maintenance process. And finally, I want to say that this is a very important thing to think about, because if you want to build brain-computer interfaces that people can use to kind of decode the complex information from their brain, if you're not able to, like, you're probably not going to use something that's like fMRI, you're going to use something like MEG. And if that information is not present in the signal, it's going to be a big problem if you want to do anything that's a bit complicated. Does anyone have any question here? I see there's some, I'm not sure.
I've got what might be a relatively simple question, but so in the MEG pipeline, I'm assuming you're doing something like a component, like an ICA component breakdown and getting localized sources. Is that correct? Or are you just streaming off of the individual squids?
Yeah, so there's no source localization in this case.
So then, but then do you think that might explain some of the complications and the limitations for your MEG results? Given that, you know, you had clean localization for superword meaning in the bold signal.
Yeah,
that's kind of blending things together with the MEG by not doing a source separation.
It's a good question. I don't think this is the case, because I would, like, you would still need to kind of, like I don't think it can be like totally not present and then appear once you do the source localization. It must be there somehow, or in some transformation of it, so it should be. But this is definitely something we're going to just verify, just to make sure 100%. But I don't think that should be the problem.
Yeah, because then at least be kind of an interesting thing why the two different signals, you know, the magnetic signal versus the hemodynamic signal, are driving two separate abilities to kind of test the encoding model, or two different outputs when you test the encoding models.
Right. Yeah, definitely. I think that's it's kind of good to kind of vary these techniques to yeah. And I think it's also interesting that people have not found this before, because I think that most people do not use MEG or even fMRI to study something that's being integrated over a period of time, unless it's like kind of a very clear controlled experiment. So So if you're not looking for these kinds of processes, because most experiments are not designed to do that, it might be easy to kind of miss the. Okay.
I'm wondering if you can comment sometime in the talk about the impact of the performance measures you choose, because a lot of these inference, especially of relative importance, is driven by the performance measure, So I don't know if correlation is an ideal one. I think there's been some debate about using it as a performance measure when you're regressing. And what would be an ideal performance measure in this context, So if you can comment on that.
Yeah, for sure. Yeah. I mean, totally, it's a totally fair point. And I must say that I don't think of any of these things as like one rule to follow, or I think there's always, our techniques are themselves a research topic that we are interested in. So correlation, I think, is attractive for like it's kind of simple to explain. It's very common in things like neuroscience or psychology, and it's easy to understand, and it's also a bit more forgiving than something like percentage of variance explained or R squared. It's just and there's like, just by saying forgiving, it's understandable why you would want that, because these kinds of signals are actually quite noisy, especially, I mean, as you know, especially MEG, but even fMRI. Ideally, when you're using a regression, I think, maybe using R squared is perhaps the preferred way to go. But we see, and there's other things you can do as well, like, for example, a classification task on top of your predictions that allows you to distinguish different things. And I think, honestly, from experimenting a lot with all of these techniques, as well as other things like statistical independence tests, like the Hilbert-Schmidt independence criterion, for example, that's also a test you can incorporate in this pipeline and replace all the kind of function fitting as well. From experimenting with all of these, we can see that actually the results are going to be robust. The pattern of results, not the values, but the pattern of results are pretty robust across all of these techniques. And so then the value of like which one you choose becomes perhaps the one that gives you more power. And again, this is a trade-off. You don't want to just increase your power, but kind of make the task trivially easy. It's a complex question. I'm not sure what the perfect metric is, but some of them, are just more forgiving than others, I would say. Okay. Any other question? Okay. So I'm not sure how much time we have left. Also.
I think on the original timeline about 10 minutes, but we have some flexibility.
Okay. Okay, cool. So, I'm going to talk to you about another way that we can do these kind of, this idea of like computational controls when we do something like this, which have a lot of inherent correlation. And this time it's not going to be about language processing, but about vision. And going back to this idea of kind of studying the effect of tasks on a specific representation, or more generally, maybe of a goal on a representation. So here in this work with my student Arya and Michael Tarr, Arya is also co-advised with Mike. We're studying basically how representations that are taken from computer vision neural networks that are trained for different tasks, how do they predict the brain differently? And just to give you a background, usually when we see these results that kind of compare CNNs and brain activity, those CNNs are often trained on a task of classifying objects in the image. And so you could ask, I mean, because that's what they're doing, then basically what they're going to do is you're going to learn a representation of the input that is optimal for classifying images or classifying objects in the image. And naturally, you would think that's not the only thing our brain can do or does. And so maybe just using these kinds of networks is not the best thing. You can think of basically using other types of tasks to do that. And in the field of computer vision, people do use these networks for many tasks. And for a while, they have ignored this idea of comparing basically the representations that you get from training the same network on different tasks. And so here in this work, which I think won the best paper award at CVPR, the authors take the same architecture, and so it's basically like an encoder decoder architecture. And so the encoder part is always going to be the same for all of these tasks. And the decoder is going to be different because all of these tasks are a bit different. So for example, some of them you input an image and then you get something like a 2D segmentation, or like you trace the 2D edges or 3D edges. Or you get you have to estimate the camera position or pose or something like that. So once you so all of these outputs are different, but still the encoder for all of these tasks is the same. And the idea is that, the question that these authors ask is whether you could use the concept of transfer learning. So basically training a network on one of those tasks and then seeing how well the performance of this network on another task. So using these measures of transferability, basically, can you actually trace a relationship between all of the tasks, or more like a taxonomy? And because it's a taxonomy of tasks, it's called taxonomy. So the idea is you take the same network, you train it on all of these tasks, and then you fix the network based, the first part of the network, or the encoder, you fix it. And then you train decoders for all the other tasks, and you look at basically the relationship between the tasks in terms of how well you are able to train on other tasks. And so you have basically then a source task and a target task in this tree or in this graph. And the idea is that the source task, for it to be able to be useful, for its representation to be useful for this target task, the information that's useful, that's needed for the target task needs to be there in the representation of the source task, and it needs to be accessible for the algorithm. Okay, so we're gonna basically use this idea of transferring from from a network trained for a specific goal to another task. But now we're going to change the task. It's not going to be one of those. So we're going to take the networks that were trained on those tasks, but we're going to try to predict the brain. And so here we use two data sets. One is the Bolt 5000 data set from Mike's lab, where subjects looked at basically images, natural images. And we also have the NST, the natural scene data set, that also is of subjects looking at real images, and it's a larger data set. And what we're going to do is similar to what I've been talking about before. So we're going to learn or train a predictive model that takes as input the encoding. For each image, it takes as input basically the representation of that image in the network. I think it's the last. So if you take the encoder part, it's basically the last layer of that encoder. And so for each one of those networks trained on different tasks, it's going to produce a representation for that image that is of the same size. And so, for example, here we take the network that's trained on object classification, and we use the representation for each image to build a predictive model. And this is the predicted prediction performance. And what we're looking at is actually a flattened representation of the brain, where this is the regions that are over here are kind of early visual cortex. So this is V1, and then V2 will be kind of parallel to it. V3 will be higher, and these kind of isolated blobs over here, these are high-level visual regions like RSC and PPA and things like that. And this is fMRI data, and so getting a correlation that's up or close to 0.5 is actually pretty good. Like these are pretty high levels in general. And so as we knew from other work, like if you take a network that is trained to predict objects or to classify objects in the image, then you can actually predict most of the visual cortex. But what's interesting is when we look at other at networks trained for other things. So this particular, I'm going to show you the results of two networks. One of them is trained to predict or to output the 2D edges of an image, and the other one is is trained to predict or output the 3D edges of the image. And so regions here that are in white are well predicted by the representations of both networks, and regions that are in red are well predicted by the 3D edges. Not by the 3D edges, but by the representation of the network that's trained to predict the 3D edges. So the 3D edges, you can go back to basically the example here. So the 2D edges are basically all the edges in the image, and the 3D edges are kind of a more high level, more involved. Even though they are simpler, they are more high level, more difficult to estimate because they represent 3D knowledge about the scene. And we see that they are actually also, more predictive of the high level visual areas, whereas the more low level primary visual cortex is actually well predicted by both 3D and the 2D network representations. And this was one subject. We can see this actually across different subjects very reliably. And we can see this also in the other data set, the Bold 5000 data set. And this is the 2D versus 3D edges. The parameters are not exactly the same between those two. The thresholds are a bit different, and also the SNR is probably different between the data sets. But you can see again that here we have the actual high-level ROI trace, and you can see that those are better predicted by the 3D voxels, by the 3D information or the 3D representations and not the 2D representations. But you don't have to end here. You can take basically all of the tasks that you have, and you can take the accuracy for each task across voxels, and you can use this to compute basically a similarity measure between all pairs of tasks. And you can use it also to create a taskonomy. So the taskonomy on the top is the one that was originally published by Zamir et al. Using the transfer learning between tasks. And what we do here is we kind of do the same thing, but we use not the transfer learning performance, but the brain prediction performance as our way to cluster and to basically build this tree between tasks. And we find kind of more like some similarities. Specifically, the biggest one is that the object and scene classification are like grouped together and different from everything else. We also see more generally a division between 2D and 3D tasks with other tasks that were not necessarily 2D or 3D, but just resemble more 2D and 3D clustering in those two. Okay. So in conclusion, we saw that we could use this kind of taskonomy or like the training networks on different tasks to try to study different parts of the visual system based on what kind of information they are presenting. And we also see that basically the main division or the most interesting comparison or the most interesting result we find that's very consistent across subjects and across data set is that specifically 3D tasks recruits the high-level visual cortex to a larger degree than the 2D tasks. And again, the main idea is that you can use this kind of correspondence between tasks and brain activity or brain activity prediction performance to infer the relationship between the tasks. So this was the second thing I wanted to mention. Any questions about that?
I think it's really interesting. I think I'm a little lost on the details necessarily about how you linked the neural network patterns to the activation responses. And is it following a similar logic as the prior encoding models that you did with language?
Yeah.
Or, okay.
Yes, exactly. It's just, so it's the same network architecture. I give too many details earlier that were not necessary. So you can think of the same network architecture that's trained to accomplish all of these tasks. But this different training objective is going to make the network learn different parameters and therefore compute different representations for each input image. And so now for each task and for each image, you can produce a vector that represents the features of that image according to that task. And so now you have the ability to compute predictive or encoding models the same way we were doing before, using the different tasks. And you can compute prediction performance again for each task, because you can think of each task as just providing you with a different encoding model.
Mm-hmm. Okay, that makes a lot more sense.
I actually have another broader question. You know, I think I feel like I'm probably questioning the work too much, but I'm just curious to find out. So this seems like the results are based on CNNs, right? The taxonomy results you just presented. How do they change if I use just say random forest or something with less capacity? You know,
sorry, I didn't hear. If you use what?
So if I switch the CNN model in this in the whole workflow with, let's say, just a simple random forest, how does the result change? You know, how can we justify CNN, not just because they were originally inspired by brain? You know.
I see. So the most attractive part about the CNN is that it's easy to kind of extract the representation of the input in a way, and the CNN is able to learn kind of patterns. I mean, because it was inspired by the brain, it is able to learn patterns that take advantage of the spatial structure of the image, and that also correspond more or less to the spatial structure that we propose, or we assume is present in the brain. Now, if you were used to use a different model, you could still, I guess, you could still extract representations from something like the random forest to represent the image, but it would just be much more like you still need like to build a model that's just as good as CNNs are currently with language, and that are that good at representing what is relevant in an image. You would need, you need to have the ability in the architecture to learn these features. It doesn't have to be a CNN, it could be another model, but the model just has to be rich enough to know how to capture like something like low frequency pattern in the image, or to treat like pixels that are next to each other in a specific way. So you need to have a model that can extract good features from the image. It doesn't necessarily have to be CNN. In fact, other architectures have been proposed that can also classify images, like transformers, for example. But it just needs to be something complex enough to extract enough information that's relevant from the image. Yeah, I would say this is the most like why you would use a CNN is just because of its ability to learn the representation. It's not necessarily a belief that the CNN is the one and only structure that could be used here.
Thanks.
Okay, anyone else has a question?
Actually, Valentina has a comment on the chat. Do you want to mention that, Valentina? Or?
Hi, sorry, I'm such a slow typer that arrived very late, but I was just saying that the comparison between MEG and fMRI, like I find it so interesting in and of itself, and it's clear that we're still lacking a lot of information, and we need more of these kind of comparisons. In particular, it reminded me of the observation that some semantic effects, like categorical effects, have been disentangled from more perceptual one in one of the two signal, like in fMRI signal, but not in the MEG signal. So it's clear that whatever we're measuring, whatever feature of these representations and property of these computations we're measuring with the two instruments is not necessarily the same, like it doesn't transpose. So I love to see more of these comparisons. I think that's a very interesting venue.
Oh yeah, definitely something to discuss further, Valentina. Yeah.
I have a question, and just wanted to. We're a little bit over our time, so I don't want to detract too much. If you had.
We don't have to cover everything. I can, but go ahead, or I can.
So one of the, the focus of the brain hack here this year is kind of on the issue of confounds and denoising. So I wonder if you could speak a little bit to, so far everything we've been seeing has been focused on the kind of extraction of the task patterns or the representational patterns and their use in kind of identifying different representational brain areas for different things. But clearly there's an aspect of kind of accounting for noise and whatnot in the analysis. Do you what kind of things do you try to use to kind of clean up your data? Because the signal to noise, and especially in fMRI, is so weak, and then Meg probably has his own issues. So what kind of issues have you, or what kind of things have you found to help deal with noise, or what are some things that you think we should be doing but we're not, that could help clean up the signals?
Yeah, that's a good question. Let's see. First of all, the stimulus, engaging stimulus is always very good when you want good responses that are clean. So having somebody, for example, read a book or watch a movie in a scanner or look at complex images is likely to lead to more engagement and therefore better accuracy or better signal to noise ratio. The quantity of data per subject is, I think, also quite important. I think that, of course, we want to run basically our models on as much data as possible, as well as many people as possible. Like, we want to kind of collect a very representative sample of the population, like we want to collect data from a lot of subjects, but if we collect only a little bit of data from each subject, we are going to necessarily not be able to estimate, have very good accuracy per subject. And then when we just rely on the fact that we have many subjects to combine our data set together, then when we do that, we are kind of basically hurting ourselves, because we can only look at that time. If we have a lot of subjects from which we have a little bit of data, we can only combine them into a smooth version of what the real underlying thing we're looking at is, a spatially smooth function. So, or spatially smooth understanding of what's going on. So we do need, whenever we want to study something complex like this, we need a lot of data per participant, and we need, and I really do believe that once you get enough data from each participant, you start to see very reproducible patterns across people. Like the variations a lot of the variation can be explained at that point by just small changes in anatomy. And I think that's quite important. I would say those two things are the most important for me. I think if you remove those, then it becomes really hard to fix any problem in the signal to noise ratio that's due to, yeah. And I can see that.
That's a good point. I think normally when we think about denoising or confounding, those issues, we often don't think about the design, and design issues, I think, are critical at kind of addressing this as well, that we often overlook.
Yeah, exactly. And I mean, yeah, again, in a perfect world, you just want, as much data as you want from as many people as you want. But I think probably the field is moving into both directions at the same time, having a lot of people and also having a lot from a few subjects. I can see that more people from the Neuromod project are here too. I also have maybe one controversial question, which I feel like a lot of the pre-processing, maybe other people who are here can maybe ask. I have a question for the audience, I guess. I think that the pre-processing and things like fMRI is not so much about SNR as much as it is about just spatial alignments. I don't think it necessarily benefits fMRI. MEG might be different, but in fMRI, I don't think it benefits the signal to, like, if the participant is not moving, I think the pre-processing's point, in my understanding, or what I think the value in it is to just for interpretation and be able to especially when you look at individual results at the individual subject level. I think that the value of pre-processing is mostly for interpretation and mapping to structural locations, not so much for SNR. But maybe I'm, I don't know. I don't know if people disagree. Maybe detrending, perhaps that's an important one, but otherwise, I don't know. That's my mind.
That is a very controversial statement. I
know.
like that, Bob. You threw it in there at the end. I think there are other things. One of the projects we're working on, at least my lab, is the physiological noise confounds, and there are ways that tasks can evoke physiological responses that can be aliased into the bold signal that like responses and things like that. But those aren't common in pre-processing pipelines. So I think you're right that like the kind of standard pipelines are optimized for blobology and blob mapping, right? Yeah. With very little kind of attention to dynamics of the task-related bold response itself. And I also feel like we started seeing the papers coming out with, slight variability in pre-processing pipeline leads to dramatic different results across, groups, all analyzing the same data set. And I think, I don't know, I feel like people's pre-processing pipelines have developed a lot like, there's a children's book called Stuck, where this little boy gets his kite stuck in the tree, and then he throws his shoe, and then he throws his other shoe, and then he throws his cat, and then he like throws a house, and then he throws a boat, and then he throws, and the whole book gets silly because he has a ladder, he throws a ladder, and in the end he throws enough stuff, and when his kite pops out, and then he's done and he leaves. I feel like that's what a lot of pre-processing pipelines get developed is, people keep adding things to kind of try to optimize their signal noise, and eventually what they get out is just like, one small piece, or a lot of times it leads to artifacts that have meaningful maps that look good. Mm-hmm. It's just kind of this additive process to pipelines without a kind of a strong attention as to what the logic is or what the reason is for kind of using those pre-processing steps. I think that's a very valid point, but.
I also agree with the first thing you said, is that I was talking about the standard pre-processing pipeline. Yeah. I haven't experimented with the more targeted ones. I'm sure that there's a lot to say here. Yeah, I was also trying to be a bit more controversial, but I think maybe people are sorry, I didn't mean to. Yeah, I am. Oh, go ahead. Sorry.
No, I want to quickly mention a lot of work from my postdoc advisor who's been pursuing this for probably like 15 years. He's of the opinion that we must optimize our pre-processing pipelines towards the final goal and question we're trying to answer, rather than just correcting for everything just because you think that you're removing something or reducing the effect of something, So just put a screenshot into the chat from one of their papers talking about how they basically compare the standard, what is the industry standard, so to say, of pre-processing with fixed optimization as well as individual subject-wise optimizations for some of the tasks. And it is very controversial. He's been telling me for some time that not many people in the FMI world have been very supportive of this work in a way. They just want to be told that, oh, apply this method, you don't have to worry about anything. That's unfortunately the what's the word, status quo.
So we're about 15 minutes over, and we still have to kind of close some things up. So Leila, if you have like 3 or 5 minutes that you want to kind of close up.
Yeah, for sure. Yeah, thanks everyone for staying on long, even though we went overboard. And I want to mention just, I don't have to go into those, but what the other project I wanted to talk about is about basically another approach in which we modeled both this was a word reading experiment that was associated with a task. And in that project, we kind of tried to model both the task and the word, as well as the task and the word jointly, in order to see which model is best predictive of brain activity. Like just thinking of some processing applied to the task and some processing applied to the stimulus or the word, and think of them as basically additive or or maybe try to learn some sort of a relationship or a interaction model between those. So this was a recent paper at NeurIPS this year. I also wanted to, oh, sorry, 1 second. Another paper I wanted to mention is a paper again with my student Maria, in which we basically found that if it was kind of a nice proof of concept that if you kind of change a network so that in this case we were actually kind of breaking the network in some way. If you change the network so that it's more predictive of brain activity, does that mean that that network is better at its task? And here this is a language network, and so we saw that like kind of the first proof of concept that kind of trying to make a network more like the brain resulted in a network that was better for NLP tasks. Yeah, I just want to finish by saying that basically moving forward, I really think that one thing we can do is we can kind of basically design full end-to-end model that start from the input and really end at the brain activity. And so this would allow us, instead of kind of taking these models that are trained on some corpora and then building a model on top of that to fit them to the brain, you could like right away learn what the features that are important for the brain are from trying to predict the brain activity right away, given enough data. And hopefully this will allow you to both kind of model the brain better, but also build at some point better artificial systems for solving the same problems that the brain knows how to solve already. And so that was it. I'd like to thank you for listening and basically thank all my collaborators, including my students, Maria and Arya, and as well as other collaborators from outside my lab. Thanks.
Thank you, Leila. Does anybody have any last minute questions for Leila? Or want to jump back into the argument that maybe we shouldn't do as much pre-processing as we were doing? I think we've got a bunch of skeptics in the audience already, so I think you were preaching to the choir. All right. Well, if there are no questions, unless do we see something pop up the chat? No. Okay. If there are no questions, thank you very much, Leila. That's always amazing work. For
inviting me. I really enjoyed it.
Of course. For anybody interested in Leila's work, I'll send a link and so you can check out her website. Now moving forward, we have another about hour of open hack, and then we wanted to converge back on at 5 o'clock to kind of go over where we are with the projects. Is that correct, Pradeep?
Yeah, so I also want to quickly bring up Isil. She's a representative from the Global Brain Hack. I think she has something to say about some of the initiatives they're pursuing. Come on, Isil, I want to put you on the spot.
But my camera will not work.
It's all right.
I'm so sorry. Okay, so thank you, Leila. Thank you, Timothy, and all, and obviously Pradeep. Thank you so much for all this amazing event and talk and all. I'm Ishil Begum from University of Reading, UK. So I am one of the members of the Brain Hack Global Organization, so we have been so lucky to have 11 different events this year, even though the year came with quite a lot of difficulties itself already with the pandemic and all the pressures put on all of us. And it's the year end, I know quite a lot of grant applications and paper submissions and going on. And so it was a huge thing to ask for to anyone to organize anything, just even a small gathering, and seeing those brain hacks are happening all around the world is kind of quite pleasing, and literally we are so proud of this happened this year. So well, what Pradeep was mentioning actually, so we have a kind of a short video about it. So hopefully if you can find a time to show you all at some point, maybe you might get a better idea. So I don't have the video with me at the moment. It's just to share with you. Well, can I do it? Or do we have time to do that? So it's kind of 15-minute long, but I will leave it to Pradeep. So one thing, the main thing that as the Brain Hack Global, we always aimed to bring the community all together and provide some opportunities to work collaboratively. It doesn't matter which seniority or background you are from, which country or university you are from. We always wanted to provide environments to make this happen for anyone, to support the collaborations and information exchange and learn from each other and support each other. This was always the main aim from the first day Brain Hack was established. And this year we kind of came up with the point that why we are not creating our own and free and openly accessible journal. So anyone who puts those efforts, it doesn't have to be the paper about a coding project or the complex neuroscience project, but it might be something you achieved through those 3 days or even about bringing an event together, the aspects you had struggled with. So something, an output that would give an incentive for your efforts you put through all those organization aspects or the project works, etc. So we thought, why we are not creating our own community venue for that, and we created this Brain Hack Proceedings platform. So it's still in the making. So I might put the links up here so that we have a public channel, it's a kind of discussion channel. Still we are creating the websites for it and all the initiatives for it together to bring something, have contributions from all the Brain Hack community. So simply, as I said, it will be free, publicly open platform that will let anyone to submit a paper, see all the review process and going openly through the GitHub issues. And anyone can be reviewer or anyone can be editor from the community, just being volunteering, just wanting to be volunteer and committing to this community-based platform is enough for us. And it's still in the making, so we are open to any contributions might come from anyone. It even has a project card on our website, so you might see it as a Brain Hack project and start contributing, starting from the issues we have in our repo. And we will publicly officially announce it hopefully next year when we have a little bit more body to it. But this was the kind of the great news and the product that we are so excited as the Brain Hack family. And we asked Pradeep to give us a kind of a couple of minutes to make this announcement and share this announcement with you. So thank you so much for that. And yeah, that's it from me to just do it without more hijacking your event.
Thank you, Issel.
Thank you.
So thank you, Leila, again. That was a fantastic talk. So much to think about, and it's really fascinating. And I was also going to ask, actually, like, how can we use this to real applications in terms of measuring language impairment or language processing impairment for biomarkers? Generally, like, if some representation in their brain is different from what is typically expected, so that's, yeah.
That's definitely a great goal. It's just understandably much more challenging because you need the right population and enough data from each participant, but definitely that's a great future goal.
Yeah, happy to work with you on that if there is an interest.
Yeah, I'll take you up on that.
Cool. Right, so Tim, we come back here at 5 or so, or?
Yeah, we'll just meet back here at the Zoom at 5 and do a project check-in.
Great. Thanks,
All right.
everyone.
See you, everyone.